on plant pigments. This is the seventh webinar in the series. So, what are plant pigments? Plant pigments are colored substances which are produced by the plant. It can be in the root, it can be in the leaves, it can be in the flower, or any part of the plant. And these pigments, the word pigment means color. So these pigments actually are chemical compounds which absorb visible radiation. And they have the color because they absorb visible radiation between 380 to 300 to 760 nanometers in the electromagnetic spectrum. And because they absorb the visible radiation, they are considered as pigments. There are many different kinds of plant pigments, and they are found in different classes, as you will see, like the carotenes, the xanthophylls, the phycobilins, the bixins, etc. And these pigments give color to either the leaves or the flowers or the fruit or the root, as in the case of turmeric. And the major pigment in the plants which is seen is important for controlling photosynthesis, growth, and development. Plant pigments, several of them are used as additives for coloring in food. These are natural colors, and so therefore they are preferred over the synthetic colors that are permitted in the food industry. FSSAI permits several synthetic colors we will see at the end, but in the natural colors are permitted in foods, but they are regulated in certain foods. So in certain foods, both natural colors as well as synthetic colors are not permitted. So there are two types of pigments in the plants, and they are known as the primary pigment and the accessory pigment. The primary pigment is the actual color that you see, and it overtakes all other colors in that particular organelle of the plant. And only with the disappearance or with the it will be a very minor constituents, or with the disappearance of the major pigment, you might see this accessory pigment as what you see in the leaves, which turn yellow, orange, or red during autumn when the chlorophyll actually fades away. Those are called accessory pigments. The major groups of the prime pigments in plants are chlorophylls, the carotenoids, and the phycobilins. And we will see each one of these, look at their structure, their function, and what color they impart in detail. So chlorophyll, this is the molecule which absorbs light. So it absorbs different packets of light called H nu, the sunlight, and it is used to convert the carbon dioxide into carbohydrates and water for the plant. So the plant uses carbon dioxide and water to synthesize carbohydrates, giving out oxygen, unlike the humans which use oxygen and give out carbon dioxide. This process is known as photosynthesis, and it is the basis for sustaining the life processes of all plants. Chlorophyll is the green coloration in the leaves. So chlorophyll itself as a molecule is a photoreceptor. That is, it is able to trap the sunlight with the most elusive of powers, because however we have tried, we cannot trap sunlight. We only can use the energy from sunlight as solar energy. But however, the chlorophyll can actually trap the energy and they are the photoreceptors of the plant. Chlorophyll is generally found in the chloroplast, which is an organelle in the cytoplasm of plants, and it makes the plant green. The basic structure of chlorophyll, as you will see, is made up of four porphyrin rings, which are coordinated to a central magnesium atom. So, if you eat plenty of green vegetables or green leafy vegetables, you will get your ample supply of magnesium. The chlorophyll structure is very similar to the oxygen carrying protein of humans, that is hemoglobin. In hemoglobin, it is held by by four imidazole rings. Iron is chelated and held, and iron is the carrier of is the central atom, and it is the carrier of oxygen. Whereas in chlorophyll, it is magnesium which is the central atom. So this is the structure of chlorophyll. You have the four porphyrin rings, 1, 2, 3, and 4, which have the central atom or coordinate the magnesium 2 plus. There are two types of chlorophyll A and chlorophyll B, and they differ only in the R structure here, where chlorophyll A has a methyl group and chlorophyll B has an aldehyde group. The rest of the molecule is, are, is exactly identical. But what you would like to observe over here is these alternating double bonds and single bonds. It is these alternating double bonds and single bonds which make chlorophyll absorb light in the visible region and give it the green color. So this explains that there are two types of chlorophyll, as I've explained to you, A and B. They differ in the R group, and chlorophyll A has the methyl group and B has the aldehyde group. These chlorophylls are very effective, both A and B are very effective photoreceptors. They also contain this network of alternative single and double bonds, and the orbitals can be delocalized. Such delocalized polyenes, that is, those which have alternating single and double bonds, have very strong absorption bands in the visible region of the spectrum, allowing the plant to absorb the energy from the sunlight. You will see that most of the plant pigments have got these alternative single and double bonds, which give them their particular color. So if you look at the absorption spectra of chlorophyll A and B, you see that both A and B complement each other. So if you look in the region of 580 to about 700, you see chlorophyll A has a very high absorption maxima, whereas chlorophyll B doesn't have. But however, in the lower wavelength, around about 600, maybe 580 to 640, chlorophyll B has got a very high absorption maximum. So the different side groups of the two chlorophylls, that is the methyl group and the aldehyde group, are the ones which tune the absorption spectra to different wavelengths, so that light that is not absorbed by chlorophyll A, say at 460 nanometers, will easily be captured by chlorophyll B. So if you look at this, so at 460 nanometers, if it is not captured by A, you will see that it is captured by B, because it also has an absorption maxima between 460 and 480 nanometers. These two kinds of chlorophyll complement each other, and plants obtain all their energy requirements from the blue and red parts of the spectrum. So here you also see that in the spectral region between 500 to 600, very little light is absorbed. So if we go back and you see in the spectral region 5 to 600, neither chlorophyll A nor chlorophyll B absorb any of the visible light from the spectrum. This light is in the green region of the spectrum, and since it is reflected, the reason is that the plants appear green. They absorb energy from the violet blue light, and they reflect green light, giving plants their green color. So chlorophyll absorbs so strongly that it masks all the lesser intense colors. Chlorophyll can be damaged when you heat or cook the vegetables, and you will see that the green color of chlorophyll disappears, and the magnesium atom in the chlorophyll is replaced by the proton from water, and that is why you see the green color disappearing. This affects the energy levels within the molecule, causing its absorption spectra to alter. The cooked leaves change color, often becoming paler, insipidly yellow-green, or sometimes even brown. In autumn, or as the days go by, in India, in the southern part of India, we do not see these changes, but however, in the northern part and in Jammu and Kashmir, you see the changes in the season, and you see that the leaves will be changing in color. So in autumn, the chlorophyll in the leaves will decay, the green color will fade away, and it is replaced by the oranges and red carotenoids, which are the accessory pigments which are present in leaves, but because of the overwhelming nature of chlorophyll are not visible. So now coming to the accessory pigments, these accessory pigments are the reason that leaves change color in autumn. In green leaves, there is so much of chlorophyll that it masks the other pigment. In autumn, however, as the tree prepares for senescence, that is when the leaves are going to fall off, the chlorophyll molecules break down, revealing the other pigments such as red, yellow, and orange that are often seen in the autumn leaves. The second group of pigments that are important are the carotenoids. These are plant pigments, and they are valuable sources not only for their color, but they are known to be potent antioxidants. There are several hundred types of carotenoids in the plant kingdom. It is not possible for us to study all the carotenoids, but let us look at the major carotenoids which have a biological significance either as antioxidants or to give color, which can be used as natural color. So the carotenoids of importance are beta carotene, which is known as provitamin A, xanthophyll, lycopene in tomato, lutein and zeaxanthin, which are very popular. Lutein especially being given for age-related macular diseases of the eye, especially due to glaucoma or to diabetes mellitus. These carotenoids range in color from red, orange, and yellow. And the familiar carotenoid, as you see, is the orange color of carrot because it contains beta carotene. So carotenoids, as such, are lipophilic pigments. That means they are not soluble in water. They are found in the membrane structures, in the chromoplasts, as the name suggests, the color, and also in the chloroplasts and in flower petals. The carotenoids can be divided into two classes. One class is the carotenes, which do not contain oxygen and so therefore are completely lipophilic. And the other group is the xanthophylls, which contain oxygen and are slightly soluble. These pigments range from yellow to orange, like beta carotene in carrots and citrus fruit, to red, the lycopene in tomatoes. Most common carotene is lutein found in the chloroplasts. The pigments primarily absorb in the blue wavelengths, allowing the longer wavelengths to be scattered and therefore producing the yellow color. In autumn foliage, the carotenoids are left over in the chloroplast and seed. So looking at the color, the absorbed color and the color observed will be different. So here is a table which tells you why one has a green color or why one has a yellow color, because it absorbs the complementary color. So you see, what you see as yellow green, yellow or orange are the ones which absorb the violet color, the blue color, or the green blue color. Now, we looked at the color of chlorophyll. So, chlorophyll absorbs in the yellow-orange region, that is 500 to 600. And so, you see, you get a green-blue color. So, to see a violet color, it will absorb yellow, green color. Now, carotenoids are mainly known not for their color, they are used as color, but however, they are very prone to fading by light, because light in the presence of light, they lose their color. But however, they are known as for their role as antioxidants. That is because they are able to scavenge the free radicals, which are unstable oxygen molecules, and these free radicals damage our body cells. So carotenoids such as beta carotene, xanthophylls are given or used as nutraceuticals, as antioxidants, especially now to fight cancer, different types of cancer. Researchers have shown that consuming beta carotene or lycopene from the tomato, along with vitamin C or E, which are two other very rich antioxidants, they can actually clear the adverse side effects of radiation therapy and chemotherapy. So, along with the along with the treatment given for cancer, for therapeutic purposes, they are given large amounts of antioxidants, and also is asked to eat a large amount of tomato. So this is the structure of beta carotene. Beta carotene is a cyclic carotene. It is obtained by the dimerization of all trans retinol. I don't know if you recollect the structure of retinol when we studied vitamin A in the class on vitamin A. So here it is a cyclic carotene. It is made up of two molecules of retinol, and by cleaving it exactly in the center, it you get beta carotene. Beta carotene has got a strongly colored red orange pigment because it absorbs the light in the region of 400 to 500, that is the blue region, so it appears as red and orange. If we go back and look at this table, you see that that which absorbs in the blue and the green blue region, ranging from about 435 nanometers to 490 nanometers, or rather 500 nanometers, 500 nanometers, you will see that, see, from 400 to 500 nanometers, it absorbs light, and it gives you the orange color. Beta carotene is known as provitamin A because when you consume beta carotene, it's sometimes it's a misnomer. People say you eat carrot, it contains vitamin A. Actually, carrot does not contain vitamin A. Plants do not synthesize vitamin A, but however, they contain beta carotene, which is provitamin A. And in the body, the provitamin A carotenoid is cleaved by the enzyme carotenoid dioxygenase to give you retinol, which is the aldehyde form of vitamin A, or it is vitamin A. So beta carotene can be cleaved to give you two molecules of vitamin A in the presence of the enzyme dioxygenase, that carotene dioxygenase. And retinol later in the body is reduced to from the aldehyde to the alcohol CH2OH, and that is retinol, retinol, and this is further oxidized to give you retinoic acid, that is, you have COOH. So these are the structures to show you. This is beta carotene, where beta carotene gets cleaved to give you retinol. Retinol is reduced and you get retinaldehyde, and retinaldehyde can be oxidized to give you all trans retinoic acid or be converted into 9-cis retinoic acid. These four are the forms of vitamin A, and this is provitamin A beta carotene, which also gives you the orange color, which can be used as a natural color for coloring food. However, beta carotene is very sensitive to light. The orange color of beta carotene, again, as I said, is due to the alternating large number of alternating single and double bonds, which allow this molecule to absorb light from the electromagnetic spectrum. So beta carotene content in different fruits and vegetables. Carrot contains the highest. You see spinach and kale, although they look green, have a large amount of beta carotene, as well as the broccoli and different vegetables and fruits have got the beta carotene content. Another color which belongs to the carotenoid family, which is used as a natural coloring agent and which is permitted by the Food Safety and Standards Authority of India, is apocarotenol. This is a carotenoid which is you mainly find in citrus fruit and vegetables like spinach. And like beta carotene, apocarotenol can be transformed into vitamin A. Apocarotenol gives you or contributes colors such as yellow to orange, red hues. And this apocarotenol, compared to beta carotene, is much more heat stable. But however, like beta carotene, it degrades on exposure to light. So it is not stable to light. And if you need to retain the packaging material of the food has to be able to prevent the entry of light. Lycopene is the major pigment in tomato. Again, like beta carotene, it is lipophilic. It is an unsaturated carotenoid. So what you see over here again is the alternating double and single bonds in repeated units. And this is the reason that it can absorb light from the electromagnetic spectrum. So in addition to tomatoes, you can find lycopene in watermelon, papaya, red grapefruits, and things. This lycopene is a symmetrical tetrapene made up of 8 isoprene units. So on this side is given isoprene unit. So you see it's made up of 8 isoprene units. However, although it's got this isoprene or long lipophilic chain like beta carotene, lycopene has no vitamin A activity and it cannot be converted into vitamin A. And it is very useful as an antioxidant, and lycopene also is a very well-known nutraceutical. Now coming to the xanthophylls, which are carotenoids that contain oxygen, you see we looked at beta carotene, apocarotenol, and lycopene. These molecules did not have oxygen. Now we look at a carotenoid which has got oxygen in it, and the carotenoids in them are beta-cryptoxanthin, lutein, and zeaxanthin. Again, lutein and zeaxanthin are the most important xanthophylls because they are antioxidants and they play a vital role in preventing the age-related macular disorder, that is in the eye, especially due to diabetes mellitus, due to stress, and they are able to scavenge the oxygen radical. Lutein and zeaxanthin are isomers. The only difference between lutein and xanthan is where the double bond is placed. So in lutein it is here, and in zeaxanthin it is down. So lutein is synthesized only by plants, and like other xanthophylls is found in high quantities, especially in green leafy vegetables such as spinach, coriander leaf, kale, and yellow carrots. It's isomeric with zeaxanthin, and they can be interconverted. The presence of this long chromophore with the conjugated double bonds or the polyene chain is the one which gives it its light absorbing properties, and it is susceptible to oxidative degradation. Lutein absorbs blue light and therefore appears yellow at low concentrations and orange red at high concentrations. So again, if we go back to this table to look at thing, you see if it absorbs blue light, the observed color will be yellow, and the absorption maxima will be around about 435 to 480 nanometers. Okay, and then we have another xanthophyll, which is cataxanthin, canthaxanthin. This also is allowed as a natural color in foods, and it is a pigment which is found in the mushroom, which is the yellow chanterelle mushroom, and it is often used to color foods, especially in beverages, and it is used as a dietary supplement. This like the other carotenoids, has this alternating units of the conjugated polyene, allowing it to absorb light, and it has the dione part of it, and so therefore it is a keto carotenoid, and this is the absorption spectra of thing. So you see it absorbs between, the absorption maxima is around about 480 nanometers, and so therefore it appears more on the orange side. Then we have a color which is annatto, and annatto is allowed in the food industry, especially to be added to butter and other compounds to give it, other foods to give it the yellow color. Annatto, the two pigments from annatto that are permitted as natural coloring agents are bixin and norbixin. So annatto is a carotenoid which is extracted from the seeds of the plant, which is known as bixa or elena, or this is known as the lipstick tree. This fruit has got a lot of spines on it, but inside the fruit you have a large number of seeds. The plant is native to South America. It is grown in India and, of course, in East Africa also. Initially, annatto is extracted in a form which is known as bixin. Bixin is highly hydrophobic, and it is therefore it is in an oil-soluble form of annatto, and this is what is extracted from the seeds. However, to make it useful in hydrophilic, to use it in foods which contain water, bixin is saponified and it liberates the dicarboxylic acid, and you get a water-soluble color which is known as norbixin. Both bixin and norbixin give the yellow color. And because you can make norbixin from bixin, it prevents the use. You don't need to use emulsifiers or gums for the uniform distribution of the yellow color in hydrophilic foods. So this is a unique attribute of annatto in the carotenoid family. So annatinoid extracts are available as water soluble, that is the norbixin, and they are available as the fat soluble as bixin. And so here what you can see is the bright yellow color, and it is added in butter. So to make even buffalo butter look like cow butter. People add the yellow color. So it is a colorant which is used in butter, margarine, cheese, fat, cereals, baked goods, yogurts, drinks, etc. Then you have capsanthin, which the name itself suggests comes from the chili or the capsicum. This is a yellow, is a red color, which is 3,3-dihydroxycarotene 6-one, and it is the main pigment. It is not the pungent principle. The pungent principle is capsaicin. However, the pigment, the red pigment that you find in the chili or the paprika oleoresin is that of capsanthin. This capsanthin has one end, if you see the structure, it has one end which has a cyclized ring, there, beta ring, and the other end has a 5-membered kappa ring over there. It has 2 hydroxyl groups. It has 2 hydroxyl groups, one over here and one over here, attached to 3 and 3 dash of the two rings and thing, and the carbonyl double bond, and the number of conjugated double bonds is 11, and you know conjugated double bonds allows it to absorb visible light, and so therefore you have the color. Capsanthin also is used as an antioxidant because it is prevents or suppresses hydroperoxide formation and has got radical scavenging activities. So most of these plant pigments, especially the carotenoids, they have got these radical scavenging property, and so therefore they are used or they find wide applications and are very popular as nutraceuticals. So the two pigments that you can get in paprika and thing are capsanthin, and the other one is capsorubin. So if you look at how these carotenoids are synthesized in the plant, the pathway, they have a very common pathway. So initially, all the biosynthesis carotenoids in capsicum, you see, have got a common pathway. So they start with the isopentenyl pyrophosphate, and they go to give you phytoene, and this biosynthesis takes place in the chromoplast. Now it is after this phytoene, which is the common precursor, you have the other molecules being formed. So in the chloroplast, the phytoene gets converted to lycopene, lycopene gets converted to gamma carotene, then to beta carotene, zeaxanthin. Now from zeaxanthin you can get either capsanthin or you get violaxanthin and capsorubin. So the path that is taken, the biosynthetic part is taken, depends on the availability of the precursor molecule. Then come to saffron, the most expensive spice in the world, and which is widely used not only as a coloring agent, but it is also used as a flavoring agent. Actually, saffron, what we get is the dried stigma. So those of you who are botany students will know that in a plant we have the reproductive parts, that is the anthers, the androecium, and the gynoecium, which is the female reproductive organ of the plant, and saffron is the stigma of the plant Crocus sativus flowers. Its golden yellow color, when you put it into water or into milk, comes from the carotenoid diglucoside, which is the diglucoside of the carotenoid quercetin. Again, look at the structure of quercetin. It has got this conjugated diene over here, which gives it the property of absorbing light in the visible region. So the major color of saffron is due to the carotenoid diglucoside alpha-crocin, which is a digentiobioside. That means it has got 2 sugar molecules here, and the basic fundamental unit of that is diapocarotene acid or quercetin. The sugar portion actually, when it is a glycoside is added over here, this is a highly hydrophobic molecule, not soluble, and when it is present with the sugar, it allows it to be soluble in water. So, if you look at saffron, the saffron flowers, it is the stigma or the gynoecium which is harvested. It is very expensive because 1 kilogram of harvested fresh flowers, there are 2,170 flowers or 2,170 gynoecium, and for the processing of every 78 kilograms of fresh flowers, you get 1 kilogram of dried saffron, which explains to you why saffron is so expensive. So here is the structure of prosin, which shows you the sugar molecules. According to ISO, you have standards for saffron, and you have got 4 standards, that is 4 grades, 1, 2, 3, and 4. So this is given by the lambda max. So the absorbance, crocin specific absorbance score at 440 nanometers. If it is greater than 190, that is the highest quality, it is grade 1. If it is between 150 and 190, it is 2. 110 to 150 is 3, and 80 to 110 is grade 4. So saffron is very expensive because of that. Because of the expensive nature of saffron, saffron tends to often get adulterated, where they create the saffron strands, or they take maize tendrils, color it with yellow orange, and sell it as saffron. So the biosynthesis of saffron occurs from the zeaxanthin. Zeaxanthin is converted, is broken down into 2 molecules. One is quercetin and the other is picrosin. And then a sugar is added to quercetin, and you get crocin. Now, picrosin in saffron is responsible, or it is the bitter principle of saffron. So picrosin and saffronol are the bitter principles of saffron, and this is the structure of picrosin, which is a bitter glucoside, which is responsible for the flavor. Picrosin is the glycoside of saffronol, and when saffron is dried after its harvest, the heat combined with the enzymic action splits picrosin to give you glucose and the free saffronol molecule. And saffronol is a volatile oil which has got a distinct aroma. Then we come to, we have now finished looking at the carotenoids, the different carotenoids and their properties, I think. Now let us look at the pigments, which are known as anthocyanins. These anthocyanins are colors, are pigments which give the varied hue of colors to the berries, that is the blueberries, the elderberry, cranberries, strawberries, etc. These belong to a group of a class of molecules which are phenols, and they are known as the anthocyanins. And they belong to the subclass of what are called as the flavonoids. Generally, most of the anthocyanins exist as bound to sugar molecules to or as glycosides to make them water soluble. So the aglycon portion, that means the portion without the sugar, is called as the anthocyanidin, and an anthocyanidin plus the sugar is known as an anthocyanin. Once again, these anthocyanins, rather than their color, they are known more as nutraceuticals. Their use is as nutraceuticals because they are powerful antioxidants, and they also exhibit anti inflammatory agent. They are used as food colorants in the beverage and the baking industry, especially when you want to get a blue color or a pink color. But they are highly prized commodities because of the value of the anthocyanins. So, among the common flavonoids which give color, you have quercetin. So, quercetin is present in a large number of products, including grapes and the skin of onion, catechin, which is found in tea, cyanidin, diazdin, lutein, and hesperethrin. These are all the aglycon portions of the anthocyanin. So, owing to the large, again, due to these alternating single and the conjugated double bond form, which carries a positive charge, these are intensely colored under acidic conditions. The lambda max in the visible region is between 460 and 465 and about 550 nanometers, and because of the phenyl rings that they also have an absorption maximum at 280 nanometers in the UV region. So this is in the ultraviolet region. So therefore, ultraviolet region is not colored. So the different types of anthocyanins known are cyanidin, peonidin, pelargonidin, malvidin, delphinidin, and petunidin. So it all depends on the R group. So their backbone is the same. They have a different R1 and R2, and the R3 is the sugar molecule or the glycoside. So cyanidin and peonidin give the magenta color, pelargonidin gives the red color, malvidin and delphinidin are purple in color, along with petunidin. And these anthocyanins, as I said, in acid, they change color because of that positive charge on the anthocyanin. And so therefore you see they are also used as indicators in as pH indicators. So here are the anthocyanins extracted from red cabbage, and you see the hue of colors at different pH. So they are red or pink at pH less than 7. They are purple to blue in neutral solutions. They are greenish yellow in alkaline, greenish yellow in alkaline solution. So this is the acidic side, this is the neutral side, and this is the alkaline side, where the pigment is completely reduced. So then beta-cyanins and beta-xanthins are collectively called the betalains, and these are the ones which contribute to the purple to blue color in berries, and especially in jamun. Also you have the beta-cyanins, and you also have the purple to blue color in the sugar beet. So they replace anthocyanin pigments. They are derived, the backbone of this is beta-lamic acid, not that of a flavonoid as you see in anthocyanins, and these accumulate in the vacuoles of the vegetative and reproductive thing. They also have got very high antioxidant activity, and the betalains from red beetroot contain about 0.5 grams of the pigment, and this is exploited for the red color, reddish purple color to be added to foods. So beta-cyanin is betaine from beetroot. So you have beta-lamic acid added to dopa, that is dihydroxyphenylalanine, and the glycoside molecule with glucose makes it water soluble. And you have beta-xanthin, which is attached to an amino acid. Both the pigments, that is the red and the yellow pigment, are thermolabile. That means they get degraded by heat, they are degraded by light, They are susceptible to pH, metal ions, and enzyme activities, which restrict their use in food color. Because if you need a food color, it must be stable to light, it must be stable to heat, and stable to pH changes. Phycobilins, these are the pigments which are present in the algae, and the word phycobilin comes from the meaning phycos is algae and bilis is bile. These are chromophores, or the light capturing molecules of the cyanobacteria. That means the colored bacteria which are there in the chloroplasts of the red algae. They are unique among the photosynthetic pigments in that these phycobilins are not free but will be available with water-soluble proteins known as biliproteins. And the proteins pass the light energy to the chlorophylls for photosynthesis. They are efficient at absorbing red, orange, and yellow and green light, but they do not absorb as well as chlorophyll A. Yeah, as you know, all the algae you find in the shallow waters, these are seen in the shallow waters, and these are responsible for sometimes when you look at these waters, you will see that instead of being blue, they are yellow or red. So they have a fluorescence at a particular wavelength and are therefore used as chemical tags in research, especially in the case of the antibody antigen reaction. There are four major phycobilins that occur in nature. You have phycocyanobilin, and you have phycoerythrobilin, you have phycoerythrin. These usually have their absorption bands in the range of 500 to 670, with a very small band in the UV region, that is below 300 nanometers. Then we come to a very well-known pigment also, which is very thoroughly studied, known to have all the properties possible, the most powerful and the most popular nutraceutical, and that is curcumin. So curcumin is the most important curcuminoid in the most popular Indian spice, turmeric. Curcuminoids are actually polyphenols. So you see here they have the benzene ring with a hydroxyl group. So here it is OH. Here you have a dimethoxy group. And from the turmeric, you can isolate the curcumins. There are three types of curcumins, that is curcumin, dimethoxycurcumin, and bis-dimethoxycurcumin. Curcumin as such can exist either as the enol form, that is OH, or it can exist as the double bond, that is the keto form. So, okay. So, curcumin, as you look at the structure, it's very hydrophobic, and so therefore curcumin is insoluble in water and ether, but soluble in ethanol and propene glycol. Again, curcumin, to make it water soluble and more effective for bioavailability, researchers have tried to synthesize the glycosides of curcumin. It is reddish brown at pH 7 and it is yellow at pH 2.5. It has a melting point in the range of 179 to 182. So this color change of reddish brown and yellow has in our Indian tradition seen very well. So you see that at any time when arti is being done with the flame, turmeric powder is added, and then a little bit lime, that is calcium hydroxide, is added to it. It becomes red in color, and only then the arti or the puja is performed. So many of you have to think of these everyday changes and things that you see. So if you look at the curcuminoids, the turmeric contains only about 5 to 10% curcuminoids. Among the curcuminoids, curcumin accounts for 77%, dimethoxycurcumin accounts for 17%, and bis-dimethoxycurcumin accounts for 3%. But of the total percentage of turmeric, you find that curcumin only accounts for 2 to 3% of the total weight of a turmeric root. The following natural colors, that is curcumin, riboflavin. We have seen riboflavin, which is vitamin B2, beta carotene, annatto extract, beta apo8 carotenoid, methyl ester of beta apo8 carotenoic acid, and the ethyl ester and canthaxanthin are the natural colors which are regulated in certain food categories by FSSA, because they do not want the natural color to be colored to be masked by the addition of these colors. So if you find chlorophyll is added to, say, pista nuts, that is not compliant, because in pista nuts it is supposed to be absent of all color. So chlorophyll is generally added to certain compounds to make them appear more aesthetic and for the organoleptic properties. So to complete this webinar, the artificial colors which are allowed in the food industry, and when I take the regulations, I will tell you about the thing, is different blues and greens are allowed, that is brilliant blue, indigotine, fast green, erythrocene, allura red, tartrazine, sunset yellow, and in India, ponzu also is allowed to think. But however, the limits are there for the addition of these colors. So that finishes the class on pigments. And so tomorrow's class will be on nutraceuticals and health supplements, where you might find a repetition of all these, because beta carotene is a nutraceutical, lutein is a nutraceutical, curcumin is a very powerful or very popular nutraceutical. So a number of these pigments as antioxidants are used as nutraceuticals.